As we have heard today, we have we will have a first speaker. So I'm delighted to welcome Poulami Somanya Ganguly, who will be speaking first, talking about mathematical modeling of stochastic processes in biology. Poulami is a postdoctoral researcher working in mathematical models of cancer evolution together with Dr. Winnie Wong, who is our 2025 Mary Cartwright Lecturer. They are in the School of Mathematical Sciences, Queen Mary University of London. Prior to joining Queen Mary in 2023, Poulami completed her PhD in applied mathematics at Leiden University and then worked at the CWI, or Centrum Wiskunde en Informatica, in Amsterdam. There she developed sparsity-based optimization algorithms for inverse problem in computational imaging and biology. So I'm welcome Poulami to the stage. The floor is yours. We will have some time for question at the end. Please do post them in the chat so that we can then come back to it. Poulami.
Thank you, Sarah. I'm just going to share my screen. Can everyone see my screen okay?
Yes.
So thank you for the really kind introduction, and I'm very happy to be prepared to be presenting the supporting lecture for this year's Mary Cartwright Lecturer Winnie Huang. So as was said in the introduction, I'll be talking about mathematical modeling of stochastic processes in biology. So this is just an outline of my talk. So I will start by introducing what I mean by stochastic modeling in biology, give some examples, and then go on to talk about how cancer evolution can be modeled as a stochastic process. I will be talking about it in very sort of simple terms because this is intended to be more of an introduction for Winnie's talk, which will talk about our current research. I will also focus on another kind of stochastic process in biology that is quite important, which is gene expression. And then I'm going to very briefly present some ongoing work on modeling gene expression in cancer cell populations. So try and bringing these two things together. So just by way of introduction, mathematical modeling has quite an important role to play in biology, and it's used across different biological scales. And for illustration, I've put two different scales in biology that I will be talking about and that are particularly important and amenable to mathematical modeling. One of these scales is really at the subcellular level in biology, so really looking at processes that are going on inside the cell. And some examples of such processes are gene expression, gene regulation, the cell cycle itself. On the other end, we have a scale which looks at biological processes from the perspective of processes happening on the scale of populations. And within this way of looking, the population could either be a population of cells. It could also be populations of different species. So we have a kind of rather larger scale and a smaller scale. And the interesting thing about the modeling, the mathematical modeling that goes into both of these scales is that often stochastic processes are important in the way that we model across these different scales. So just by way of, again, very introductory remarks, a simple kind of stochastic process is the linear birth-death process, where essentially we look at a population and we can think of individuals in that population either giving birth or reproducing or dying. And this simple process can be modeled either deterministically or stochastically. So in the deterministic setting, we would write down the time evolution of the numbers of individuals in the population And then we'll have an equation like this, where lambda is basically the birth rate and mu is the death rate. And we can write down this differential equation and solve it exactly, and we get this kind of exponential growth or decay given the relative weights of lambda and mu. In a stochastic setting, we would instead model them using a language of, so I've shown them here as kind of chemical reactions, where you have the birth process in the first line and the death process in the second line. And we would assume that these birth and deaths events happen independently and randomly. When we solve this system, then deterministically, we would expect only a single trajectory for certain parameter values. However, in the stochastic case, we would expect there to be fluctuations around this deterministic solution. So what we see is that in biology, these fluctuations are important. They're not only there in real biological systems, but also, for example, in this process, we would never exactly expect the number of, we would expect this to sort of asymptote towards zero, but never really reach zero. So there wouldn't be any perfect extinction. But in the stochastic processes, we would get a distribution of extinction times. So for these properties, we would have to move beyond purely deterministic modeling and towards stochastic modeling. So cancer itself can be modeled as a stochastic evolutionary process. So there are various theories at the present moment as to how cancer is initiated and how cancer advances. One of the theories is the somatic mutation theory, which postulates that cancer is a disease driven by accumulation of so-called driver mutations. So in this picture, if we have, for example, a population of cells, and we call these somatic cells. So these healthy somatic cells are here shown in these pink cells. And then we have a cell that carries a certain mutation M. The question then is, what is the probability that the mutation that is carried in this cell will become fixed in the entire population? And probably predictably, one way to kind of model this is by modeling it as a birth-death process. To make kind of matters simpler, in a very simple situation, we could imagine that the population of cells is constant, so that the births and the deaths at any given time are happening sort of like as if one birth happens, then one death will happen at the same time. So I've kind of shown this process by circling the cell. So at every time point t, this birth-death process in a constant population can be modeled as following. So we pick a certain cell for reproduction, and the cell here that's circled in green, and we also pick another cell for death, which is circled in red. So essentially, going from time t to time t+1, we have a replacement of one cell by another. Again, at time t+1, we'd go through the same process. What's important to realize that it's possible to select the same type of cells. So for example, it's possible to select cells of type S, both of them for birth and death, which means that going from time t+1 to t+2, you wouldn't see any change. So in this kind of modeling cancer, this birth-death process in constant populations, we could further look at a specific case, which is known as neutral drift. So neutral drift, the idea is that it is equally likely for a cell carrying a mutation to replace an unmutated S cell as it is for an S cell to replace an M cell, so that there is no specific advantage for carrying this particular mutation. In this neutral drift case, we would then write down the transition probabilities of going from having small m cells of type M, and we would model this, given that it is a Markov process, we would only be able to go from small m cells to either m-1 cells of type M or m+1 cells of type M. And given that we are in a constant population, by definition, we have capital N, which is the total size of the population, minus M cells, which are of type S, so the healthy somatic cells. And so we can write down these transition probabilities. We essentially have three situations, either M going to M minus 1 and M going to M plus 1. In this particular neutral drift scenario, these probabilities, these transition probabilities are the same. And the probability of M remaining at M is obviously 1 minus these two terms. So the fixation probability, which is what I talked about in the previous slide. So the fixation probability is essentially the probability that starting at M equals 1, so when we only have one cell with a certain mutation, then the system will end up in a state where M is equal to capital N, where capital N is the entire population size, and this can happen in any number of steps. So we designate this probability as rho 1, and we can expand it in terms of these transition probabilities, where P1 of 0 is the probability of moving from M is equal to 1 to M is equal to 0, and rho 0 is the probability that is the fixation probability when we have M is equal to 0. So here, it's important to realize that when we have M is equal to 0, then that is a stationary state of the system, because you could only ever choose the same kind of somatic cell for reproduction and death. So that rho 0 is essentially 0. If we expand this, I will not go through this in detail, but however, if we expand this kind of recursive relation, we get that rho 1 is half of rho 2 is 2/3 of rho 3, etc., until we get to rho is, for rho n minus 1, I'm sorry, apologies, there's a typo here. So when rho n minus 1 is n minus 1 divided by n, rho n. Rho n again, so the state where we already start out with the whole population as being the mutant cell, again, that is a stationary state. So rho n is 1. So we get an expression for our fixation probability in this kind of simple scenario, which goes as 1 over n, so that for smaller populations, this fixation probability is larger. A more sort of realistic scenario to do the same kind of modeling is to assume that there is some advantage that the cell has for carrying this particular mutation. So usually in cancer, these kinds of mutations would either give the cell some kind of proliferative, it's assumed that it would give it some proliferative advantage, or it would suppress cell death. So in mathematically modeling that process, we would talk about the concept of fitness. So fitness here is essentially the reproductive success of a particular M cell. And under the assumption of constant selection, we would assume that the reproductive success of a cell carrying this mutation is some constant S times that of the healthy somatic cells, so that when S is greater than 1, we say that the mutation M is under positive selection. If it is less than 1, we say it's under negative selection. And if it is equal to 1, then we're back in the neutral drift case that we've just seen. If we incorporate selection, then the probabilities, the transition probability of moving from M to M minus 1 and M to M plus 1 are no longer equal as in the neutral drift case. However, we can write them down. And again, P of the transition probability of remaining at state M is given by 1 minus these two terms. So kind of repeating the same kind of calculation as before, we can again get an expression for the fixation probability. In this case, the fixation probability, that starting from one cell carrying the mutation, we get to a state where the entire population carries the mutation, is not only dependent on the size, but it's also dependent on this selection fitness, so on this selection coefficient, which is S. One of the things to note is that because we are modeling this stochastically, even in the situation where n goes to infinity, where you would have the fixation probability as just being given by 1 minus 1 over S, you still have a finite probability of there not being fixation, so that there is no guarantee of fixation as such. So this is a very kind of simple setup and a very simple example of how cancer is modeled in using stochastic processes. Wayne in her talk will talk about more sort of realistic examples of how this has been done by other postgraduate researchers in the group. So I will move on to introducing something else that will be relevant to when we sort of go into Wayne's talk. And that is again, a biological sort of concept of extrachromosomal carriers of oncogenes. So extrachromosomal DNA or ecDNA. So far in the picture that I was showing you, the mutation copy number, so by that I mean that how many genes carry this mutation and how that mutation is then inherited at birth, so at cell division, they implicitly followed the statistics of chromosomal, so-called chromosomal inheritance, where essentially what that means is that one mutated gene would be replicated at cell division and then segregated evenly among the daughter cells during cell division, so that we expect that if a cell carrying a mutation divides, then both the daughter cells would then inherit that mutation in the same way. This is true if this mutation is carried in the chromosome. However, there have been recent observations that in certain types of cancer, there are genetic mutations which are carried not only on the chromosome, which in this schematic we can see as marked as X, but also in so-called extrachromosomal DNA, which are essentially circular particles of DNA, and they do not have the machinery that ensures that the amounts of genetic material that get passed on to the daughter cells at cell division are equal. So what happens during cell division if a cell carries extrachromosomal DNA is that they get segregated randomly into the daughter cells, so that now we have different possible outcomes at cell division. So for example, if a cell starts out with 2 copies of extrachromosomal DNA, then at cell division, these 2 copies would replicate to form 4 copies. And then these 4 copies could be segregated randomly, so that we can have one possibility is that only one daughter cell inherits all the 4 copies. And then you have other possibilities where you have a certain fraction of the copies going to one cell or the other. The way that we mathematically model that is that we say due to random segregation, the oncogenes, the mutated oncogenes, are going to be inherited by daughter cells according to a binomial distribution, where n, which is equal to 2i, is essentially the number of extrachromosomal DNA that we have at cell division, and we choose k of those to go into one daughter cell with probability 1/2, and the other 2i minus k copies go to the other daughter cell. In this situation then, the kinds of questions that are asked is, what is the probability that the entire population, so similar to what we were addressing before with the kind of simpler case of mutations, what is the probability that the entire population of cells will contain the oncogene if it is carried on ecDNA and segregated according to this random process? And what is the probability distribution, moreover, of the ecDNA copy numbers that are there in each cell? Because now we will not have all of the cells having the same numbers of ecDNA. So the results of this are going to be part of Wayne's talk. I will move on slightly to why we sort of want to then move beyond simply looking at genetic composition and genetic number, both in the context of biological systems as a whole, but also in the context of cancer. So far, we have assumed that there is a 1:1 relationship between the genetic composition, so between the fact of whether a cell is carrying a mutation or not, which is in biological terms would be its genotype, and some observable characteristics, for example, the rate of cell proliferation or its treatment response if it is a cancerous cell, so which would be in biological terms called its phenotype. However, it has been observed in different model systems. So here I'm showing a population of bacterial cells. So it has been observed that even cells that are genetically identical can be phenotypically diverse. So this is a population of bacterial cells that are expressing the same gene. So they are producing the same protein. They are under the same environment. These genes are identically regulated. However, the way that each of these bacterial cells fluoresces is different, which suggests that there is some inherently random process that is not simply captured if we assume a 1:1 relationship between carrying a gene and its observable characteristic. Importantly, in the context of cancer cells as well, when a certain kind of protein, which confers the cell with a kind of proliferative advantage, when this certain kind of protein is labeled fluorescently, so what you see here is a group of cancer cells, in particular glioblastoma cells, and some of them fluoresce as yellow, which means they are high in a certain quantity of protein, and some of them fluoresce as red. And these two different kind of cell subpopulations, they have different properties when treatment is administered. So in the cells that express this protein to a high level, there is a high percentage of cell death, so that they are actually amenable to therapy, whereas the ones that do not show this expression are not. And if we simply measure the copies of ecDNA, the copies of extrachromosomal DNA, which carry this particular gene, what is noticed is that even in the cells that seem to show a low expression, we seem to have a high number of extrachromosomal DNA. So that simply carrying the gene does not coincide with showing a particular observable characteristic. So this makes me move on to like gene expression as a stochastic process. So gene expression as a whole is the entire process of going from genotype to phenotype, of reading out genetic information to produce some observable characteristics. We will only focus on the first or on one of the steps of that process, which is transcription, which is the production of so-called messenger RNA or mRNA from DNA. So the way to kind of mathematically model that, again, is to assume that this is a stochastic process. And the simplest way that gives biologically relevant characteristics is to assume that it's possible for a gene to exist in two different states, so that a gene could either exist in an activated state or an inactivated state, and only in the active state it is able to produce mRNA. While if a gene were always transcribing mRNA continuously, we would expect a Poisson distribution. If we allow for this gene switching, what is observed is that mRNA is only produced in transcriptional bursts, so that if we have a distribution of the mRNA number, then it tends to have a long tail. And in experimental measurements of mammalian cells, it's been observed that actually this long tail is observed, so that the experimental distribution does not fit the kind of Poisson distribution which we would expect if only one gene were transcribing mRNA continuously. So to write this down again in the form of like stochastic chemical reactions, we would have a few steps. So we'd write this gene activation and inactivation as a step of going from G to G star with certain rates. So the rate of activation is Ka and the rate of deactivation here is Kd. Then we'd assume that when the gene is active, it produces mRNA with a certain rate Jm, and the mRNA are able to decay with a certain rate Km into some neutral decay product. We can model this as a continuous, so here, instead of taking a discrete space, we usually model this in continuous space. So we have X, which is the normalized mRNA concentration, which can go between 0 and 1. And for simplicity, we assume that the active gene always transcribes to mRNA. so that this bit is deterministic, so that the only bit that we care about, we have these kind of rescaled activation and deactivation rates, which are the rates Ka and Kd normalized by the rate of mRNA decay Km. So we can write this down in terms of a Fokker-Planck equation. So we would write down 2, so we can have the gene as existing in either the 0 state, which is inactive, or the 1 state, which is active. And P here is the probability that we would have a certain concentration of mRNA X at a time t when the gene is inactive, and P1 when the gene is active. So we have a few terms. The first term for P0 is the term that corresponds to mRNA decay, because when the gene is inactive, that's the only thing that's happening. When the gene is active, then we have 2 terms. We have a term that stands for mRNA production, and a term that stands for mRNA decay. And furthermore, we have some gene switching terms, where we have the rate of activation and deactivation entering. So for the case of like just where there's only one copy, it's possible to solve these equations for the steady state. So the steady state probability distribution function of the mRNA concentration X will be a sum, obviously, of these two, P0 and P1, and the steady state. In the steady state, we get a form like this, where we have these two rescaled activation rates, RA and RD. And N is the normalization constant. So for different regimes, which we will call slow and fast switching, in such a system, when one gene copy in each cell is expressing stochastically, we would expect different behaviors. So for the case of fast switching, where these two parameters, RA and RD, are greater than 1, so that the gene is switching much faster than mRNA can decay, the mRNA has a chance to accumulate. So here I've kind of shown this with these colors of these cells. So for the probability distribution function of X, we would assume that we would expect that this would peak somewhere in between at 0.5. So that we have essentially most of the cells in the population will show some kind of mRNA concentration that is close to the average mRNA concentration. If RA and RD are different to each other, so this is the case for RA equals RD, if RA is smaller than RD, then we would have this peak much closer to 0, and the opposite, much closer to 1, if the activation is much higher. The more interesting case, when we have slow switching, when RA and RD are less than 1, then we would have a bistable or a bimodal distribution, where it would be possible for the cell to either have very low mRNA content or very high. And this is the case when, with the same population, with the genetically identical population, we would still be able to get stable subpopulations of cells where the properties of the cells could vary. And again, this is for RA equals RD. If RA is smaller than RD, we would only get one peak towards 0. And when RA is much greater than RD, then we would expect that peak to be close to 1. So in our situation with extrachromosomal DNA, given that extrachromosomal DNA segregate randomly at cell division, we will take the analysis that we've done for like one copy, and we'll try to extend that to when we have different numbers of copies in our cell. So to do that, we don't do it analytically, we will solve it numerically. So now, instead of each cell just containing one copy of a gene on extrachromosomal DNA, we will assume that there is instead a distribution of possible copies. And we will solve the transcription problem. So we will simulate transcription using the Gillespie algorithm, which is essentially models each of those reactions chemically, and where we, at each step, choose random numbers to decide which reaction has taken place. So we will simulate that transcription to give, for the same population, the mRNA copy numbers. And I will show some results where we extract the bulk ecDNA distribution, the bulk mRNA distribution, and where we also extract some single cell data and how we compare that to experimental data. So just by way of kind of simulation, when we numerically solve this, what we observe is that for the same ecDNA distribution, so here we have an ecDNA distribution that peaks at around 20, given that we have these different activation, deactivation rates, we are able to get different bulk mRNA distributions so that when the activation rate is much smaller than the deactivation rate, we have an mRNA distribution that is much more peaked at 0. And slowly, as the activation rate is increased, we get to a situation where the other limit case, where we have the activation rate as much higher than the deactivation rate, where we have the ecDNA and the mRNA distributions almost coinciding. For a small area of the parameter space, where our parameter space is RA and RD, for a small region of that parameter space, we also observe the kind of multi-stability that is observed in the one gene copy case. So here, when RA is equal to RD and when it is 0.1, for instance, what we observe numerically is a rather smooth distribution. However, for RA is equal to RD, when it is lesser than 1 over n, where n is the peak of the ecDNA distribution, which is around 20 for our case, we observe a regime where we see multiple peaks emerging, similar to the bistable situation, but here we have multi-stability. So transcriptional stochasticity, we can kind of simulate that, and we see differences between the bulk DNA and mRNA data, and for certain values of stochasticity, we observe distinct subpopulations. We can also do it in, we can also simulate and get the single cell data. I will not go into this in much detail, but however, what we do observe in the single cell case is that we have a variance between cells that contain the same ecDNA copy numbers, and also with kind of changing the relative values of activation and deactivation. Not only does this variance decrease, but also the slope of the linear fit moves closer to the situation where all gene copies are active. So the takeaway from this is that cells with the same ecDNA copy number will show differences in their mRNA concentration, and the variance is a function of the relative gene switching rates. How does this compare to experimental data? So from our experimental collaborators, we have some data of real genes that are expressed on ecDNA, and we have this kind of single cell data where we have the copy numbers of these ecDNA and the expression, which is the mRNA numbers. And what we see for different genes that are present on the same extrachromosomal DNA, what we observe is that the single cell kind of data for mRNA distributions and ecDNA distribution, the joint distribution essentially, is different for different genes. In our simulations, these would correspond to different values of activation and deactivation for the different genes, and we would be able to qualitatively get similar distributions to the ones that we observe experimentally. So this is just to say that qualitatively, we could recapitulate the differences in expression as essentially differences in the activation and deactivation rates. To do some more kind of quantitative comparison, what we do is we use the extrachromosomal DNA copy number distribution from our experimental data. We use our simulation pipeline to simulate the mRNA distributions, and then we can extract either bulk data so that we have just the histogram of mRNA and the cell count, or we can have some single cell, the joint distribution of mRNA and ecDNA, depending on which data we want to compare with. And to compare with experimental data, we minimize the Wasserstein distance between our predicted, so our simulated distributions, and the experimental mRNA, either counts or distributions that we have. And then we use that to pick our parameters RA and RD. In order to test this, we first do some inference on simulated data just to see that we can recover these parameters. And what we see kind of predictably is that when we do these predictions with single cell data, since we have more information, we know exactly which cells have mRNA counts equal to these ecDNA copy numbers, we can usually infer our parameters better than if we were just using the histogram. So just to show some kind of quantitative fits to the single cell data that we have. So from the single cell data, so we have them for different cell lines, and the amplicon refers to the kind of ecDNA, and essentially this is the gene name, but I think that's not very important. Essentially, what we have is we have for different genes, we will have we have data on the experimental mRNA copy numbers and the ecDNA copy numbers. And with our best fits, we kind of get quite close to these experimental distributions. What we observe, which could be like biologically which is subject to more investigation at right now, is that we can, from these fits, we see that the activation and deactivation rates they differ for different genes in the same extrachromosomal DNA. So if these extrachromosomal DNA happen to carry these different genes, we would like to understand better why we get these predictions for there being different rates. Would that mean that the geometry of the extrachromosomal DNA matters? Or how can we sort of biologically explain that? What we also observe and what we also get from our predictions is that the activation and deactivation rates are similar for the same gene in different cell lines. So in different contexts, what we observe is that if we are only observing the same gene, then the values for activation, deactivation that we get do not seem to depend on the context in which that gene is present. And so we're also interested in trying to figure out why that is. I will just end with a summary. So I introduced a little bit about how biological systems at different scales are characterized by randomness, and presented a very simple kind of birth-death process that's used to model the spread of oncogenic mutations in a cell population. Then kind of briefly looked at why mutations may not, or oncogenes, carrying oncogenes just themselves may not be able to predict the observable characteristics of cells. So we looked at a two-step stochastic model of transcription and applied it to cancer cell populations and showed qualitatively how that could recapitulate some of our observed mRNA distributions. And just as an outlook, some of our future work is to try to understand some of the predictions on the rates that we get and to incorporate additional sources of phenotypic heterogeneity, such as more complex interactions between genes. With that, I'd just like to thank you for your attention. Thank you.
Thank you very much, Poulami, for setting the stage so well and for your introductory lecture. I wonder if there are questions from the audience. As Kiran has put in the chat, you can either use the chat or you are also welcome to raise your hand and we will give you, and you can ask your question. So I wonder, let me... Okay, perhaps if no one wants, or while the audience might think about what they want to ask, let me say, it is great to see, the progress made and the variety of models. I, as a non-expert in cancer modeling, I wonder if are there types of cancers that are more amenable, say, to mathematical modeling? Is there any difference? How this very important topic can be tackled, whether you know of any particular difference in the various families?
So in the kinds of models that, particularly for extra chromosomal DNA that I showed towards the end, so there are certain cancers where this, particularly glioblastoma and particularly brain, so pediatric cancers like glioblastoma and neuroblastoma, where these extra chromosomal DNA have been observed, so that this particular kind of line of research of looking at the statistics of extra chromosomal DNA and then building on that to model the gene expression, so might be more relevant for those kinds of cancers where this is observed. Okay.
Thank you.
Yeah, if I may add, so in general, I think it depends on the disease. So maybe you heard about in general, when people have this cancer, they might test whether they have special type of genetic markers. So some cancer, for example, even in colon cancer, so some of them are rising from a lot of mutation, which I will talk a little bit about point mutation. Some of them are more like the whole chromosome is disrupted, and some of them don't share genetic similarity between patients. So then people believe there are more like what Poulami talked about, phenotype switching, like you have the same genetic background in the cell, but then you kind of changing depends on the body of different people. So that's why there are a lot of luck going on. So if you have a genetic driver inside of the treatment, then you could maybe model it in one particular way. But if you don't, then you need to model different type of stochastics in the system. So it's very important to use the right type to reflecting what is the mechanism behind if the mechanism is not known yet.
Thank you very much. Let me check. I don't see any further questions. So thank you again, Poulami, for your very nice lecture.